Okay, so objective of today's class is to get an overview of biomechanics, and our focus scope will be restricted to K. We'll just get an overview of what kinds of questions, what are there in the syllabus, what are some references that are very useful for learning this. So that's the objective of this class.
We certainly have 10 plus marks based on what is given over here. Okay, 10 plus marks is a lot, it's a big chunk. Kind of know, like vaguely where questions show up from, but let's give it a very, very concrete form so that it's useful for preparation as well as for revision. So now let's get into details of all of these things and show you actually what they mean by forces, moments, equilibrium, biomechanics of joints, what are the things in biofluids, biomaterials, and all. And so with that, now let's get into Git, right? Let's continue with the daily chores. So this is what, we are all curious, but our day-to-day life, we need to move on with things. So let's get with this thing. So what are the questions that show up from biomechanics? Let's restrict to Git so that we can constrain things to Git. What are they asking biomechanics? You have seen 2020 paper, 2021 paper. Basic resultant vector calculations. Okay. Modeling and activity. Perhaps by that you mean either you are referring to, say, some kind of degradation, tissue degradation, things like that. And then modeling this ion channels and, origin of biopotentials. That Young's modulus, of course, that fits very well with biomechanics. Things about
biology related.
Yes.
Biology related to biomechanics, like the anatomy part and some forces apply. Anatomy part they frequently ask, and anatomy and the forces applied to different organs or the position of the body, like leg, arm, and all. Mostly this two types variants, like we have seen till 2020 and 2021.
Okay. Yeah. So that's an interesting thing. The analysis of joints. So biomechanics of joints or applied statistics. Not statistics, applied statics of joints. Right. That is a very, very typical question. Pretty much you can expect every year one question from that. Then stress strain curve, application of stress strain curve. That again is Young's modulus, stress strain, applications of that, properties of stress strain curve. That is one thing. And then of course there is also fluid mechanics. Now why have this? Why discriminate solids or tissues, or like tissues that have a structure, how about tissues that flow, right? How about blood, and perhaps the tissue that where the blood is flowing, blood vessels and all that. Yeah, this is where questions typically show up from. Great. Let's give this. We kind of know, like vaguely where questions show up from, but let's give it a very, very concrete form so that it's useful for preparation as well as for revision. It's going to be an overview. It's going to be very fast paced. Yeah, but at the end of the day, you'll get a very nice structure to help you prepare, also help you revise. Okay, so first thing is, let's say what the syllabus says, kinematics of muscles and joints. Okay, basic movements of muscles and joints. So in fact, some anatomy and physiology helps here. Okay, some basics of tissues, and then musculoskeletal system in particular. Some knowledge of that, and some basics of how do you define movement in a body, locations in a body. All of that help define kinematics. That is one thing. Then we have free body diagrams and equilibrium. Essentially applied Newton's laws. Newton first, second, and third law in different forms. Okay, applied. So this is called as statics. And in biomechanics, we are interested in applying this to muscles and joints, right? We are not applying this to cars and wheels. We want to study muscles and joints. And a part of this is, of course, we want to know why we do that, what are the forces in our joints, whether the joint will withstand that force, what are the stresses in the implant we put in a joint, whether the stresses are too high so that the material will fail or not, things like that. Okay, so at the end of statics, so when we do this study, we get the results on forces and stresses typically. And yeah, that's also pretty much, biomechanical analysis of joints is essentially that, applying statistics to joints. Then we have this thing called gait analysis. We have, different ways to describe gait, what are the angles involved in different positions of the gait, phases of the gait, and what are the muscles involved in gait, all that show up in gait analysis. And then we have hard tissues. Okay, this is where, okay, now we want to model the things that we are studying about. So we have things like stresses and strains. Okay, how do bones behave? How do cortical bones behave? How do cancellous bones behave? Then we have something called soft tissues, because the stress strain characteristics of hard tissues will be very different from that of soft tissues. So again, properties of soft tissues in terms of the stress and strain. Sometimes if it falls short, we need something called viscoelasticity here. This is roughly say this is elasticity, this is viscoelasticity. And then we have biofluid mechanics. So let's get a much better picture of this. This is a bird's eye view, what is there in the syllabus. Let's get into details and look at what is there. Yeah, before that, let's go over the gate questions. Quickly say, where do you think they are from the syllabus? Which of the following is not a part of the knee joint? What kind of a question is this? We don't want like answers now. I want to know where they are, showing up from the syllabus. I feel all of them are biomechanics questions. So yeah, notice, yeah, joints. When you talk about joints, first thing is like describe a joint, anatomy, right? And then functioning of a joint, perhaps functioning of a joint, there is not a lot, but let's say anatomy and physiology. Here, there is an anatomy question. Okay, HAP. Then how about this? Thin filament of a muscle fiber is composed of. Yeah, this again is anatomy. We are talking about, just organization. We are not. So anatomy and physiology. What is anatomy? What is physiology? Anatomy is just talking about form of something. What are all the things there and all that. Okay. Physiology is talking about the function of something. Okay. So how it functions, talking about that is typically physiology. For muscle fiber, what are all there in a muscle fiber? That is anatomy. What happens during cross-bridge cycling? How does actually muscle generate a force? That will be physiology. Okay, but briefly, this is a HAP question. And notice this has to do with musculoskeletal system and specifically microstructure, because you will see this trend again in microstructure. Okay, now, okay, what about this question? Which of the following statements about smooth muscles are true? Basic, it comprises both anatomy and physiology. There are smooth muscle. What are smooth muscle cells? Where are they located? Part of that, and how are they being controlled, and how do they, work? So this has to do with HAP. And again, notice an important part of musculoskeletal system known as the smooth muscles. Okay, properties of that. Then we come to this question. A person is sitting in a chair with the feet on the ground. While rising up on his feet, the kinematic motion modifies. Oh, yeah, these are all from biomechanics. We want to be like, start pinpointing where they are showing from the syllabus. Yeah, this is known as kinematics of joints. Right? We had some kinematics of joints. So we have all these movements of joints. So kinematics. So kinematics is just the study of movements. Kinematics of joints. So all these movements are given a term so that we can communicate again. So this is pretty much the anatomy of movements. Then we also have the physiology of movements, which is where we study, free body diagram, equilibrium. Okay, how about this question? Comparing parallel fiber pinnate muscles of a given volume with other statements. Yeah, this is a interestingly, this is a combination of two things. What are pinnate muscles? What are parallel muscles? And they also ask about their function. Okay, what do pinnate muscles do? What do parallel fibers do? What are they good at? They're good at shortening, or are they good at generating force? This is a proper HAP question. But notice again, it boils down to musculoskeletal system. Most of the HAP questions, there are one or two from which gland secretes which hormone. Questions like that are there, but mostly they are all centered around musculoskeletal system. Musculoskeletal, this muscle architecture, tissue architecture, and in fact, there is both anatomy and physiology. Okay, this question: person standing position first flexes the hip by this much. So, what is this? Flexes the hip, trochanter knee ankle line, distance between hip joint and knee joint. Yeah. All these questions are from biomechanics. Yeah, again, notice this is again description of movements of joints, right? Again, this is kinematics, and specifically, I would also, this is closely related to gait analysis, very closely related. Nothing to do with terms in gait analysis, but this is your starting point of gait analysis. In a particular phase of the gait, one could always ask, okay, where are things? Locate the ankle in a particular phase of the gait. That is very likely possible from this. Kinematic question closely related to gait. Okay, how about this? 60 kg person is standing on the foot on a force plate. Ground reaction is this. Again, mass center of the foot is trochanter knee ankle. The weight of the foot is 0.8 kg. Closest magnitude of moment acting on the ankle joint is. Okay, let's read. So at the end of the day, what they want is magnitude of the moment acting on the ankle joint. So kinematics has to do with movements, but this biomechanical analysis of joints, analysis of forces, stresses in the joints. Moment is just a specific type of force. Yeah, this is applied statics on joints. You apply statics on joints. You could also call this biomechanical analysis of joints. So this is a typical numerical question you could always expect. If it is not the ankle joint, it could be the elbow joint. If not the elbow joint, it would be this pivot over here, some part of the perhaps the spine, hip. Pretty much name anything you can apply. What are the forces in it? Okay, so very important question. A question like this we cannot answer if we don't know what is trochanter. Knee ankle, perhaps we can make it this. What is trochanter? Right? If we don't know anatomy, again we will be like we cannot answer that question. And then, so again, basic kinematics. If we don't know what is flexion, what is extension, this is a very simple question at the end of the day, but we cannot answer. So all this is very, very closely linked. We need to know anatomy, at least for the musculoskeletal system, very well. We also need to know kinematics. Then we need to know basic gait analysis to make sense out of the question. Once you make sense out of the question is actually very, very easy. How about this? So novel biomaterial was tested for tensile properties, so and so. I think all of us have read this question. So I am kind of like fast forward. So based on the experimental results, this biomaterial would be suitable for bone, tendon, ligament, articular cartilage. Yeah, Swarnendu says this is deformation mechanics. In order to study forces in a joint, you think the joint, the parts of the two things in the two sides of the joint are rigid. But in order to go one step deeper, to study the stresses and strains, you need to get out of that. Rigid body doesn't suffice. That is, rigid body is something that cannot change its, that is rigid, okay, cannot change its shape. The moment you come out of that well, you generalize it, you get deformation mechanics. You can start studying about stresses, specifically strains. And this is where the Young's modulus, material model concepts and all that show up. So we have this stress strain relations, right? Material properties, all of that. Very illustrative question is this, and it's kind of given in a data interpretation format. Some table, again, a very simple question: calculate Young's modulus. That's what they say, okay? And then they kind of obfuscate that whatever that simple question into this table and this long sentences, okay? So at the end of the day, they say, what is the Young's modulus of this question? Which range does it follow? Okay, that's what they ask. Yeah, this is a question from deformation mechanics. Again, you can expect at least one question from this every day. These are very predictable. And let me highlight a couple of details at this point. Notice this is closely related to biomaterials. Now, the distinction between biomaterials, numericals, and biomechanics, numericals and biomaterials, that kind of like that boundary is like pretty much fades because you study mechanical properties of biomaterials, you're talking about biomechanics. Okay, first thing. And if you look at this, so biomechanics, we can see it's closely related to biomaterials. You could always expect a typical numerical from this, typical numerical from this. These two are very typical. Young's modulus and forces in the joints. But notice some of these anatomy questions, it's very hard to pinpoint what they will ask. Okay, something in the musculoskeletal system, something with respect to microstructure, but very hard to pinpoint. These are theory questions. But the numericals, you can easily pinpoint. This typical numerical, okay, very easily pinpointable, typical numerical. And this is also another typical numerical. Then let's go one step further. Okay, diameter of a renal artery lumen is this. Reynolds number of the blood flowing through the renal artery. Where is this showing up from? Yeah, this is biofluid mechanics. Now we are talking about mostly things with blood. Okay, cardiovascular system, intact cardiovascular system. We are talking about biofluid mechanics. Okay, questions from this is like very, I would say, typical numerical. Just like questions from these two are very typical. Again, there is something related, like stress and strain, we do something, like Young's modulus, there is something like Young's modulus and biofluids called viscosity. A typical question is expected from that. And there are some very basic properties of flow, Reynolds number, things like that, typical numerical. And let me also point out something at this point. So last year we were prepared for this. Reynolds number, everyone in the class knew what Reynolds number is, how it is applied, how to calculate it, everything. But they did not know what this CP is. Okay, what this, it says centipoise. Okay, they did not know, C is centi, but P, they did not know, P is not Pascal, unfortunately. Okay, P is poise. They did not know what that was. But they knew Reynolds number, they know how to calculate it and all that. But because of that, they did not know what poise is, what that damn unit is, just like that Curie in medical imaging, they did not know what is poise. So many lost marks here. So yeah, we have to pay some attention to units, the typical units used. Not just the SI system, but also this, typically things that show up in the books. How about this? Okay, consider blood as a suspension of RBCs in a Newtonian fluid. Shear forces experienced by RBCs during blood flow. Yeah, basic mechanics. Yeah, basic, in fact, stress, strain, biofluid mechanics. Yeah, this basic biofluids, some theory, some, can you apply some principles of biofluids to this thing? Not a recall-based question, but applying a principle, biofluid mechanics principle to the question and then answer. So this is what's shown up. This question. How about this one? Glomerulus filtration process of kidney modulus flat membrane with pores of radius 1 nanometer, length of pore 16 nanometer. Viscosity of the fluid is so and so. Average pressure of bedside membrane is this. Ultrafiltrate side is this. Total available area of membrane is 1.5 meters square. Nearest value of resulting filtration rate. This, I would always say, is a typical numerical. This is actually biofluids question. One of the basic things in biofluids. Again, even before, before the 2020 questions, whatever I say are typical, we are expecting that even before the first biomedical gate paper was out. Okay, so I had some example problems for that. So, okay, biofluids. Interestingly, this is applied to bioinstrumentation. Okay, this is biofluids applied to bioinstrumentation. Now we are also starting to see this relation. It was connected to HAP, it was connected to biomaterials. Now it's also, in fact, many designs, many measurement devices uses basic principles of mechanics, biomechanics or biofluid mechanics. Okay, in this case, it's closely related to bioinstrumentation. Okay, and finally, I have one or two more questions. Tensile strength of a degradable suture is the closest approximation of the tensile strength. Yeah, this is curve fitting, exponential curve fitting, I suppose. Yeah, decrease exponentially. That is exponential decay, find some parameters, given some perhaps at most 2 data points. Yeah, this, I said, okay, let's, there is a lot to think about this question. First is, it's actually one, short phrase, it's exponential curve fit. And we have already seen exponential curve fits can show up from any corner of the syllabus. It can be from analog electronics, it can be from biomaterials as in this case, or, as we are again talking about properties of suture, biomechanical properties, mechanical properties, it also comes under biomechanics. This, at the end of the day, is exponential curve fit. Applied, it shows up as a biomaterials problem, but at the end of the day, I would also include this in biomechanics. At least questions like this, biomechanics and biomaterials are closely tied. The origins of biopotentials, biomechanics and bioinstrumentation is closely tied. All this diffusion, Nernst-Planck equation, electrophoresis, all of that. Okay, biomechanics, close tie up with biomaterials. And if I have to name one thing as a most typical numerical, I would always name this exponential fit. Okay. So most typical numerical. I can't imagine a gate paper without a question of this type, that is exponential curve fit. Finally, what do we have? Yeah, this thing. This is temperature of bone cement, so and so happens. Coefficient of thermal expansion. Assume that. So what they want is stress developed due to the exothermic reaction, stress and polymerization process and shrinkage. Stress and strain. Yeah, this has to do with deformation mechanics. This is actually a question from biomaterials, but it makes use of this interesting material property called thermoelasticity. Again, if you understand basic stress and strain, basic biomechanics, you could easily answer this. Thermoelasticity. Yeah, biomaterial question, but underlying principle is biomechanics. Okay, with that, so this is my summary of whatever we have here. In 2020, there were close to 12 marks from biomechanics. In fact, most of the questions that I showed. 2021, close to 8 marks. Okay. So let's say on an average, at least 8 marks. 8 to, on an average, about 10 marks from this. If you put together associated HAP, biomaterials, and also bioinstrumentation, we certainly have 10 plus marks based on what is given over here. Okay, 10 plus marks is a lot, it's a big chunk. Comparable to medical imaging in that sense, because it's not just this, but also biomechanics, biomaterials, anatomy and physiology, and bioinstrumentation to some extent. All of them closely related to what is given over here. And roughly, questions from anatomy and physiology, human anatomy and physiology, typically from musculoskeletal system, something to do with skeletal joints, muscle system, muscle architecture, microstructure of bone, muscle, things like that. Then kinematics, so many examples, flexion, extension, sitting, standing, and so movements of joints, and that also involves gait analysis, slightly different. Phases of gait is very different from uh, terms we use for describing moments. Then, okay, just talking what are forces, what are moments, what is equilibrium, what are the Newton's laws and all that. That is underlying theory. When you apply it to that, we can, we showed this ankle joint analysis. Okay, that question of type of question. Tissue properties, all this stress, strain, Young's modulus, things like that. In fact, there are a couple of more things. I'll give a slightly detailed overview, but tissue properties, things related to stress and strain, typically tensile strength or Young's modulus. Okay, biofluids. Again, there are like 2 or 3 handful of things that very typically show up, and many of these concepts, tissue properties and biomaterials, are closely related. Numericals from this, or most likely expected. So, and then of course we have theory questions spread all over. Okay, so now let's get into details of all of these things and show you actually what they mean by forces, moments, equilibrium, biomechanics of joints, what are the things in biofluids, biomaterials, and all. First thing, yeah, we should always start with directional references and planes. So, what is anterior, posterior, proximal, distal? Even it's referring to even the ankle is so and so to the thing, TKA line, anterior to the line. We need to know what is anterior, basic directional terms like this. Also, the planes help us. This is where it is a reference to define all the movements. We need to know this. And then musculoskeletal system always start with the Latin roots. This will simplify our life. So instead of memorizing 1,000 words, all you have to memorize is 50 words, and then that 50 you can easily blow up into. You can easily decipher the meaning of 1,000 other words in musculoskeletal system. So always start here with Latin roots and their meanings and see how they are applied in the musculoskeletal system description. Okay. Once you understand the roots, most of this names will be like evident. Then musculoskeletal architecture. So this is body level things, anatomy. Now, how are these muscles organized at a tissue level, right? So in fact, we had questions from pinnate and parallel muscles. So here we have the anatomy, but go one step deeper, look at the physiology. What can generate more force? What can generate more contraction, right? Again, if you understand this architecture, that is, if you understand the microstructure of this tissue, you could also guess that, okay, all the sarcomeres are aligned along this direction, so contraction will happen along that way. This is likely to contract. There is this contraction has a cos theta term, so contraction is going to be lesser, but on the other hand, it can generate more force, things like that. Okay. Then basics of skeletal systems and focus on the joints. So this is pretty much the anatomy of skeletal systems, and yeah, here the microstructure of muscles is what I meant by, okay, we have the sarcomeres here. This is how we have, okay, this is composed of thick and thin filaments, which in turn are composed of actin, myosin, troponin. That anatomy part of it. So any contraction is going to happen along the length of this. So I know which direction contraction happens, right? Get a basic feel for that, and also look at the physiology. Underlying physiology here is known as cross-bridge cycling. Okay. And then we can look at the muscle microstructure. We can also look at the bone microstructure. You need to know what a cortical bone is, what a trabecular or cancellous bone is, because now we'll start describing their properties. We want to replace a bone with a material. Which material is appropriate? Right? We can start asking those questions. Typically, we'll look at mechanical properties first. So first thing is step is understand bone microstructure. Also understand the bone composition. What is a mineral component? Then does it contain water? Basic questions like that. How does water affect its mechanical properties? Okay. In fact, yeah, I'll come to that towards when we look at mechanical properties of biological systems. Then the step towards going towards physiology of joints is first understanding classification of joints. Okay, so I suppose we have classifications like this. This is fibrous, synovial, and cartilaginous, right? So understand the classification of joints. This is a starting point of physiology, and pretty much know different kinds of approximations for joints. Based on allowable movements, we classify joints into something. Of this, the most important is the hinge joint. Okay, I'll explain why this is the most important joint in a moment when we look at equilibrium, equations of equilibrium. And even if it's not the elbow, if some other joint is given, let's say ankle joint is given as in the GATE 2020 exam, we will always approximate that as a hinge. We won't consider that as a plain joint. This is for simple classification problems. If we do it as a plain joint, we cannot solve the problem. For that reason, we'll always treat it as the hinge joint is the most important joint. And I'll also explain why that is the case just in a while. Okay. And then basic kinematics. What is flexion? What is extension? Understand why this flexion. This is flexion because, okay, this angle appears to be increasing, but this is flexion. But, this angle is decreasing. This is flexion. Understand that properly. It's defined with respect to the proximal segment. So understand basic kinematics. Abduction, adduction, rotation, circumduction, basic things. Okay. Pronation, supination, dorsiflexion, plantarflexion, inversion, eversion. Yeah, we have given a nice list of all the things over here for revision, and many of these use proper anatomical terms. In fact, sometimes they can just give you the description and ask you to guess for which motion they are referring to. It's very easy if they give you the picture and tell me the motion, that's very easy. But sometimes they can give you the description, ask for the motion. So be prepared for that also. Okay. Then finally, we have this summary of gait analysis. We need to know the different phases of the gait, and then corresponding joint angles, what is inflection, whether the hip is in flexion or extension, knee is in flexion or extension, ankle is in flexion or extension. Then which muscle is actually active during gait phases. You need to know this. This is very, very basic. This we can pretty much, like, if we know what the stances are, we can pretty much guess this. This is slightly hard. We need to memorize something. Okay. And then comes statics. So why is the hinge joint most important? Let me answer this. Statics, what are the things we need to know? So we always have, like, we'll be look at 2D equilibrium, that is, what is equilibrium? Anyone can say that? Because, okay, equilibrium is given there in the syllabus, but what is equilibrium? Okay, net force is 0, Himanshu says net force is 0, fine. Yeah, equilibrium actually could mean a lot of things. It can be chemical equilibrium, it can be mechanical equilibrium, things like that. Here we are, things are stable. Yeah, equilibrium is a very, very broad term, but in biomechanics, equilibrium refers to something very, very specific. Equilibrium means no acceleration. In fact, instead of saying no acceleration, you could say the body is not moving. Body is like held fixed, because we have this Newton's 3 laws, right? We start from that. Okay. First says what is a force? Body will keep doing whatever it's doing until some external force acts upon it. The second law, in fact, says that force is actually defined by mass times acceleration. Here, notice, okay, we are not going to details of what is force, why is force, mass times acceleration, and all that. It's just one of the Newton's laws. Okay, so let's stop there. But notice, first of all, we need to know what a force is. Okay, and force is a vector, acceleration is a vector. So according to this, if a body is in equilibrium, that is, if it is not accelerating, okay, the force is, net force acting on the body is 0. We typically write the net force as summation of all the forces in the body is 0. Okay, so this comes from Newton's laws, definition of a force. And equilibrium, by that we mean acceleration is 0. Okay. So we have summation of all the forces is 0. Notice this is a vector that will have a x component, y component in 2D. In 3D, of course, there will also be a z component, but to make life easier, this sigma f equal to 0 is where we get these two relations in 2D. If you look at things in 2D, in 3D, we'll also have one more term, sigma f z is 0. Okay. So if you add all the net forces in a body, things become, if they add up to 0, that means the acceleration is 0. That means the body, or that is how mechanical equilibrium is different. Net forces acting on a body match each other. And the body should be isolated. In fact, we'll come to that in a moment. Equilibrium, we are talking about equilibrium of a body. If the body is connected to some environment that is not a body, right? Now, okay, for example, this pen, I am holding the pen, right? If I want to analyze the pen, I have to separate the pen from my hand. Okay, that aspect of separation is known as freeing a body. Depicting that is known as free body diagram. Okay, that is, in order to analyze a pen, we can only analyze all these isolated bodies, equilibrium of bodies that are isolated from the rest of the world. So the process of isolating things, the process of freeing bodies is known as, and depicting that is known as free body diagram. How to do that, I will explain with lots of examples, simple examples will build on top of each other. So the moment we have isolated a body, that is when we have a free body diagram, we can say its net acceleration is 0 for it to be in equilibrium. That means the net forces should be 0. Force is a vector. If we add all the forces, it should be 0. So where do we get this equation? Sigma m is 0.
So the equilibrium you referred here, isn't it only force equilibrium?
Okay.
Because if you have two equal and opposite forces acting at different points, say the points of action is different, then the force is 0, but the moment is not 0. So the body is typically not in equilibrium.
Yeah. In fact, so for a body to be in 2D equilibrium, the pen shouldn't be moving up or down. For example, if I drop this pen, it's accelerating in the y direction. It's not in equilibrium. If I hold it, that is, if I apply there is a gravitational force, I apply another force to, counter that, it stays without acceleration. That is in equilibrium. So it stays the same way in y direction, stays the same way in x direction without moving. There is one more requirement. The body shouldn't be rotating, right? If there is an unbalanced force, two equal and opposite forces, but if they are not, under again certain conditions, they can make the body rotate. So body shouldn't be translating, body shouldn't be rotating. The third condition gives us this. In fact, if you add all the moments acting on a body, that should be like this inertia, mass, inertia for acceleration, things moving in straight lines. There is inertia for angular acceleration, things rotating. This angular acceleration, inertia for that is typically known as I. Okay, and in 2D it can rotate in only one plane. In 3D it can rotate in 3 planes. So let's forget. In 2D we have one rotation in that plane, and that should also be not happen. So that angular acceleration should be 0. So net moments should also be 0. So these are the 3 equations that we need, we need to satisfy in order for 2D equilibrium. So that is the first thing. So we need to know what a force is. We need to know what a moment is. Of course, I will explain that. But essentially, from Newton's laws, equilibrium is the body is not accelerating either translationally or rotationally. We get these 2 equations. This is what we'll apply. If we apply this to joints, isolated joints, free body diagrams of joints, we are talking about applied statistics, biomechanical analysis joints. So now let me raise this question. why hinge joints? In fact, yeah, I might have a, yeah, let's raise this question. That is, I have shown like various joints here. So you could see what is allowed, what is not allowed on here. Why hinge joints? Can anyone look at these three equations and tell me why hinge joints? I said, they're typically not going to give you pivots for analysis, ball and sockets for analysis, condyloids for analysis. They will mostly give you hinge joints. Or even if they give you ankle, which is said as a plain joint, classified as a plain joint, you will approximate that as a hinge joint in order to solve the problem. Why is that?
Hinge joints typically allow movement in a particular direction, in one particular direction.
Okay.
So that direction can be defined as like 2D coordinates, like X and Y. In a plane, movement is happening. So that's why hinge is the most fit.
Okay, because 2D. Okay, fair answer. Net moment. Because 2D. So this is a very interesting answer. Okay, let me ask you a different question. Now we have three equations. Now, how many unknowns can we solve for? Three linear equations, sum of all forces.
Three unknowns.
Yeah. Now, if you isolate a hinge joint to draw a free body diagram, so you have to account for this. I am cutting it out from the rest of the world, right? I have to account for the mechanical effects of that joint. Hinge joint, when you isolate it, okay, it will give you three unknown forces. Why that is the case, we'll also explain. I'll explain when drawing the free body diagram. But when you isolate a hinge joint to draw a free body diagram, that is when you make a cut at the hinge joint, that will expose three unknown forces. So if you know pretty much all the other forces in the system is given, you can solve for all these three forces using three equations. Other joints, ball and socket, no, this is very, very best example is a rigid joint. Rigid joint will have like 6 unknown forces. How can you solve for 6 unknown forces with 3 equations? You can't do that. And many of these joints will run into troubles, except for this one beautiful joint that allows only one rotation in a plane. Okay, when you isolate this part of the body from where it's connected, when you make a cut here, this cut, I have to account for all the mechanical effects of whatever it's connected to. The mechanical effects will correspond to exactly 3 unknown forces. So this is the only case where we can solve for all the unknown forces using the 3 equations. The number of unknowns match the number of equations. Because of that reason, this hinge joint is very, very special. We have this statics, and then we apply that to study equilibrium of joints, that is applied statistics, equations of 2D equilibrium. Then comes stress and strains. So first question is, why study stress and strains? Why not study forces and deformations, changes in length? Why this? Anyone? Okay. Let's say if you are talking about, so here what we have here is like stress versus strain. Stress is force per unit area, that is normalized force. This is change in length per original length. Let's say original length. So changing length, we can talk about, okay, which length are we talking about. So we study this normalized force and normalized change in length instead of directly the force and length. That's because now I have a pen made up of one material. I have this pen. I study force versus change in length of this pen, and I make the pen longer, twice its original length. Then the force versus length changes will be very different for that new pen. It's made of the same material, but how force changes with length will be different. Similarly, if I make this pen thicker, the force and how that varies with the change in length, how that varies with applied force, that will be very different, although it's made up of the same pen. So for that reason, okay, let's take a pen. Let's take a longer pen. how the applied force is related to the change in length will be very different for this pen and this pen, although it's made up of the same material. Okay. You could either make it longer or you could make it thicker. That is, you can either change the length or you can change the area. For example, if I were to compare this, if I apply the same force, if I were to compare this, it's going to stretch longer. Intuitively, perhaps you could feel that. Or in this case, it's much easier. If I apply the same force to this material, it will stretch smaller, right? So, although things are made up of same materials, if I plot just the applied force to the change in length, let's say they are straight lines, but they'll be very different straight lines. To get everything to the same page, we normalize them. If we plot force versus length, we don't want 3 curves, but we want 1 curve because it's just one material. So look at the material and apply it for different geometries. Interestingly, if you plot P over A for the 3 cases over here, they will all be the same line. They won't be 3 different lines because they are made up of the same material, which is why we start studying stresses and strain. So we are specifically mechanical properties of materials. So stress, then, so the moment we start talking about stress and strains, there are different aspects in stress and strain. What they typically ask is this initial slope called the Young's modulus or the tensile strength. In fact, the tensile strength, yeah, we'll get to that. There are things called yield strength, ultimate strength. There are so many parameters here. And what is going on here? What is going on here? Will this change based on the type of material? Will it be the same for a bone? Will it differ for a soft tissue? Or even take a bone, the bone is wet, density of the bone changes, how will this changes? All of that, either just the properties here of this curve or properties of this curve apply to a specific material. Both questions are possible. Okay, we have stresses and strains. Then we have, typically when we talk about stresses and strains of soft tissues, we need viscoelasticity. Again, we are studying stresses and strains. But how are these stresses and strains different from the stress strain just drawn here? Any guesses? Okay, this at the end of the day is stress and strain. Viscosity, will come to that. Okay, again, stare at the figure that I have shown. So, in fact, let me also explain what happens here. We take some viscoelastic material. We apply a constant stress to it from T0 to T1. If you measure the strain, it's going to look like this. That is, the material will slowly start creeping. Okay, there is an instantaneous jump. Okay, but after that, as I hold the stress constant, it will slowly change its length. We'll go to equilibrate at some strain, or by the time we stop applying the stress. The moment we start applying the stress, there is again an instantaneous jump, and then this happens. So what is the key detail here? Creep deformation.
Non-linear.
Creep relaxation. In fact, if we look at this curve, it appears to be linear here, but beyond here even kind of gets non-linear, right? The relation shown here is non-linear, but there is something more interesting about this. Yeah, this is known as the creep. There is creep, and then there is stress relaxation. We'll come to that. Okay, Megha is actually very close. She said modulus of elasticity. Can you try to elaborate a little more, Megha? How is modulus of elasticity? Is the modulus of elasticity constant here? Is ratio of stress and strain a constant? Changes with time, right? That is, at this region I have this stress, I have this much strain. Same amount of stress, but I have a different amount of strain. Kind of the modulus of elasticity seems to be a function of time. Right? So the key element here are things are functions of time. Things are no more instantaneous. In this case, You apply a stress, there is a strain. You apply a strain, there is a stress instantaneously at that very instant. In fact, you could see that happening here. The moment you apply a stress here, there is a sudden jump, vertical jump, that is instantaneous. This is what we studied in the previous slide. Whether that goes linearly, non-linear, that's a different story. But there is an instantaneous response. You tell me sigma, I'll tell you strain. There is no time dependence here. Here we have this time dependence. Sigma of t is some function of epsilon of t. Very, very simple relation. It can be functions of derivatives of epsilon of t and all that. There are many ways to write this, but things are functions of time. One example is the modulus. If you look at the ratio between stress and strain, that appears to change over time. Simple thing. Okay, so things are functions of time. Functions of time. And so, in other words, this is like a zeroth order system. There is no time dependence. This is like a, what I have shown here is almost like a first order system approximation. Things exponentially saturate to something. Like a first order system is like a zeroth order system. There are second order models and so on, but that in another term, signals and systems terms is a first order system, that's a zeroth order system. Or there is time dependence here. In fact, what I have shown here is what is known as creep. That is, I hold the stress constant, look at what happens to strain. I could do the other way around. I can take a material, I can make it strain fixed, that is, stretch it and hold it that way. Measure the force that happens there. Interestingly, the forces or the stresses in the material will change over time. Okay. In fact, that is known as stress relaxation. So you can apply this and study this, or you can make this constant and study that. That is possible. Or even if this dependence of time is not evident for you, one simple example is, if I make sigma sinusoidal, sin omega t, epsilon will again be sinusoidal, okay, but with a phase difference. Just like, this is almost like having a pure resistor. This system is like having an inductor and a capacitor somewhere. So now things are functions of time, if that analogy makes better sense. So this is like a purely resistor. We are studying voltage and current in a system with pure resistance, but now here we have impedance. Things are functions of time. We introduce a phase shift between voltage and current because of the impedance, or there is time dependence. You could look at it that way. So such models are known as viscoelasticity. And again, typically soft tissues, biological materials, we have bones, tendons, cartilages, even bones. If you change the material properties, if the bone is wet, or you apply stress and strain at, based on the rate at which you apply the strain, bone properties will change. So most biological materials, viscoelasticity is an appropriate model. Okay. So things are functions of time at the end of the day. So that is viscoelasticity. Again, we study stress and strains, but as functions of time. Then biological tissues are more interesting. Yeah, one is what I have shown here is I think a slide of a bone. Okay, so biological things are mixtures of things, right? So what if I don't use one material? I have used one or more than one material. I need something called mixture models for describing stress and strain. Specifically for biomaterials, biological tissues is very, very key. Okay? Very important. And then more interestingly, if I try to, again, if you approximate all these relations using springs and dashpots, or resistors and capacitors, you will have a very, very strange element. Not just something that is dependent on time, okay? Something more stranger. In fact, I have shown that here. First of all, the stress-strain relation for, for bone, it might look like this, and then it will go somewhere like this. It will almost look like this. But if you take a rubber band and do that, if you take a soft tissue,
Dentin cartilage, it's going to almost very small stress, it's going to change a lot. Then suddenly become very, very stiff. It's going to be exponential. Okay, unlike this, it will do this kind of behavior. Okay, so how do we model that specifically for soft tissues? That exponential dependence between, say, this is force and let's say length, relative length change. Roughly, this is, let's say, stress versus strain. That exponential relation is shown here. Okay, that is a passive thing. Interestingly, the actual force you measure won't be just that. There are something in the muscles that are like motors, right? How are we able to lift our hand? Because there is almost like a motor here, right? There is something active in addition to being the active, as in there's something like a motor in the muscles. There is a contractile element that makes, if you think of this as a sarcomere, there is something that protein conformation changes, some crossbit cycling that generates a force. That's very, very new outside of living systems. You take any material, they can't generate a force of their own. Biological systems, muscles can generate force of their own. So there is also this additional active force drawn in that brown line over there. And then finally, so the net force you measure will be a combination of this active and passive force drawn by the given. So this biological tissue, specifically muscles, first of all, the model is very exponential. There is a passive component to it. And then there is also active component to it. Interestingly, this is there in the syllabus, which is why I'm talking about this. And then we have to talk about, of course, not just biological materials, but all the implants that we put inside the body, right? Metals, all the, even composites, all of that, polymers. Stress and strain, notice, actually the typical thing they ask is Young's modulus, but there is a lot more in the syllabus. And then finally, biofluids and TAMS, the other things. Biofluids, the idea is this. Okay, now we have this intact cardiovascular system. We want to study pressure, flow, perhaps stresses and strains in the system. What we typically do is we'll approximate this system, this network, as again another network, but a simpler network, where blood vessels are like straight lines or like tubes. At the end of the day, these are also tubes. We just align them and study them. Okay. And if you look at any one tube, you try to describe what is the pressure of flowing blood and all that. What is the flow rate of blood? How are they related? We get this thing. So the connection for that is we had Young's modulus for studying stress and strain in solids, right? For fluids, the equivalent of that is blood viscosity, or in fact viscosity. Okay. Here we have what is known as a Newtonian fluid. It's a straight line. What I have here is a rate of shearing strain. Shearing strain is change in length as two planes shear this way. The movement I am showing. This is elongation. Okay. This is shearing. Okay. That is, if you look at any straight line between the planes, they will change angle as the things shear. And we are looking at the rate of that shear strain, not just some strain. We are looking at shear strain and the rate of that given by that. And here we have the shearing stress. So we said sigma is A times epsilon. That relation for fluids becomes this shear stress is viscosity times the shearing strain, du by dy. Okay. Again, the strain is this. In fact, we have the rate of strain. There is a rate term here. I'll explain that later. But this is proportional to this becomes the shear stress is proportional to rate of shear strain. The proportionality constant is known as viscosity. Again, this proportionality constant, based on that, we have different fluids and the blood. What kind of fluid is blood? That is a typical question. So this is the starting point. And now we can apply this to study. I take a blood vessel, I apply pressure here, P1, P2. The kidney filtration problem, ultrafiltration, glomerular filtration, essentially makes use of this. Now, what is the flow in it? Okay. In fact, it's very important. Now I apply the same pressure in a narrow blood vessel. What should happen to the flow?
Should it increase, decrease? Can we quantify that? A simple way to study that is known as Hagen-Poiseuille or laminar flow. Analogy for this, I apply a voltage, what is the current in it? Okay. Instead of a voltage, we apply a pressure gradient, okay, and we say what is the rate of blood flow. So we have like a voltage like term, we have a current like term Q. What connects them is this resistance like term, the resistance of this blood vessel. This, in fact, we already saw a problem on this, the tissue glomerular filtration problem. Okay, so how are the applied the pressure gradient or change in pressure related to flow inside a blood vessel? That relation is extremely important, very typically expected, and this expression can be written in many ways. Okay, this is the relation. Okay, how do we get this expression? Can we, instead of flow, can we talk about velocity of blood, mean velocity, maximum velocity, basic things? So this we need to study, and at end of the day, this is essentially we have a resistance like term, current like term, and the voltage like term. And then how good is the flow? You might have, this is very easy to explain, right? Sometimes observed, if we pour something, it flows very nicely, but the moment it hits, it becomes kind of like becomes very turbulent, right? Or flowing streams, things don't appear to be very nice, right? Or things appear to be very chaotic. So to characterize that, we need something called Reynolds number. Again, these Reynolds numbers are based on the basic theory here. We use this, built upon this, based on the flow rate, velocities, we can define what the Reynolds number is. There are many ways to characterize this. I mean, these are all equivalent. So we have the viscosity here, and then mu over mu is known as the kinematic viscosity, this mu over here. Okay, so that term disappeared into this mu. Or you could start writing the velocities, mean velocities as the flow rate over area, some basic things like that. You can make some couple of conversions. Very straightforward once you know what is going on. But to characterize what kind of flow is happening, this number is very essential. It says roughly we have like mass-like terms here, okay, and we have viscous terms here. How is the inertia of the system affecting it? Ratio of that to ratio of viscosity. How viscous is the system? That ratio determines whether the flow is going to be nice or whether the flow is going to be turbulent. Okay, so Reynolds number, basic calculation of that. It's simple. Similar to whether the flow is like laminar or whether it's turbulent. Similar to that, we can say whether the flow is pulsatile or steady state. So the number we use that for that is known as the Oemersley number. So again, for intact cardiovascular system, very important non-dimensional number. So this is transient inertial forces to viscous forces. That was inertial forces to viscous forces, just the transient part, that part that changes as heart pumps. So this is another important number over here. And finally, to put things together, okay, how do we characterize flow inside blood vessels? We studied that, some properties of that flow using Reynolds number and Oemersley number. And then what happens to this blood vessel that has this flow? Will that blood vessel rupture, or there too much stresses and strains in the blood vessel? What are the properties of the blood vessel? That question we can ask. So we get back to where we started. We can talk about stresses and strains in the blood vessel, okay? Or we can talk about the material properties of the blood vessel. So here a cross section of a major artery is shown. What are different, where is the smooth muscle cell here? What are these different layers? What are the mechanical properties of that? What are the stress strain relations of that? That we can study. Or as say, given a specific pressure flow relation, what will be the stresses here? That also we can study. These are slightly advanced, but the more basic one is, the most important one is this. Everything else builds on top of the other. Okay, so this is what are there in the syllabus. Let me again, I have a summary here. By now, this should make better sense, I suppose.
basic anatomy and physiology with a focus on musculoskeletal system and microstructure. Then kinematics and gait analysis, of course, that we understand very well. Statics part, just studying forces, moments, what is equilibrium, some basic mechanics principles, statics. Applying that to biological joints systems is biomechanics of joints, biomechanical analysis of joints. And here we typically treat it as rigid bodies. Things don't change their size dimensions. Okay, but that is not all that useful, because this is an approximation. It's useful for some purposes, studying joints and all that. But to study blood vessels, muscles, and all that, things change dimension. So we need to understand stress-strain relations, whether it's viscoelastic, whether it's active, passive, and all that. Then flow of blood and pretty much in pipes, right? Intact cardiovascular systems, and of course, the composition of blood vessels, composition of muscles, and all that. So biomaterials is close to it. So this is what we have in this summary. Okay, now that the overview I gave should help you decipher what is there here. Yeah, make use of this overview to revise, to prepare, to see what is there as a roadmap. And this map is certainly not the size of the subject. Okay, this is not, everything is not right here. This is just a map for you to give you some sense of direction while you are preparing the subject. Use this as a map, as a roadmap while studying, and as a map for revision and all that. But we have a lot. What are some references we have for that? So notice, yeah, these are questions that I put up before the 2020 exam. I knew a question is going to show up from this, either the ankle joint or this elbow joint. Okay, yeah, I thought it was either the elbow joint or the ankle joint, one of the two hinge joints in the system. Yeah, one question showed up in Gate. We have a playlist for Gate biomechanics. That's a nice reference. Go take a look at it. Biofluid mechanics, certainly you don't really have any material other than whatever I put. So all the things I said, starting with, okay, what are all there in the system, brief overview on biofluids, this analogy between electrical circuits and this pipe flow, because we are used to this. So I start with that analogy, then study this pipe flow, Hagen-Poiseuille flow. How do we calculate resistance? Then can we apply that to systems, pipe flow to, say, this kidney filtration process, study what happens to shear stress, then can make some generalizations there, Bernoulli principle and all that. Is one of the best playlists. I mean, not one of the best playlists. It's pretty much the only playlist out there for public playlist for biofluids. Go take a look at this. Right now, I also curated all of them at one place. Okay, all the links, Wikipedia links, open source links at one place in this YouTube video for anatomy and physiology notes. In fact, my plan is to make such a notes available for biomechanics. This is likely to go there. And for examples, now let's look at the book reference. We need more than one book, certainly. So Enderle and Bronzino will give us an overview. Generally, biomechanics is split. It's there in one chapter or fourth chapter, pretty much all the examples say. Asgay or Norden, so the statics, applied statics, stress, strain, biological tissue. The basics. They are very well covered here. What are forces, moments? Okay, what are equations of equilibrium? How do we apply that equations of equilibrium to a rigid body, to joints? And then, okay, things are not rigid, things are deformation mechanics. What are stress and strain? Multi-axial is, it's not there in the syllabus, but just in case you have some extra time, you can look at that. And then, okay, how do we start describing things that deform, and how do we apply that to biological tissues? So that is also there in Asgay or Norden. And this chapter, biological tissue, is very, very, very closely related to 9th chapter in Cork and Gates, because it talks about biological properties, stress strain of biological materials, their composition, how do you model that? So most of basic biomechanics, the solid mechanics part of that, engineering mechanics, can be found in Asgay or Norden and Cork and Gates. And then for biofluids, we need Lee-Whiting. All the things I said, Reynolds number, Womersley number, viscosity, everything split over, I have examples from all the chapters, so this should help. And kinematics and gait analysis, we need external references. It's not there in any of these books. So again, I have good references for that. So we also have references for that. So you already have a nice list.
So with that, our count should be 28 from aptitude and math. Then roughly, at least 12 marks from medical imaging. If you combine everything, biomechanics, biomaterials, at least the basics, then HAP, all of that, you certainly have 10 plus marks. Okay. I would say even 12 marks. So whatever we do here has again a lot of marks. We'll again master it. We won't leave loose ends so that just in case this question showed up, I don't know, we didn't see this, and that thing likely won't happen. I think we'll cover things properly. Even before we pick bioinstrumentation, I am expecting that 40. You should go 50, 52 plus, and you have been practicing well. You should get 40 out of 52. Easy subjects. That's a plan. Okay, so with that, that's the plan. That's the schedule. Okay, have a good one. Bye bye.
Thank you, sir.
Thank you,
Thank you,
sir.
sir.
Thank you, sir.
Thank you, sir.